Okay, so now we'll get started on the next lecture for QBI. And this lecture will be a little bit shorter than the other ones because, as you see, many people have already left for Easter. And so we don't want to cover too much material. And the next lecture depends on this one quite a bit. And so it's important to have covered this one. So yeah, the outline remains more or less the same as before. And this week is analyzing single objects. And then, not next week, but I think the week after, we'll be analyzing complex objects. And so this will build up sort of the basis for what we do with the images once they're kind of segmented. And after that, it will go into some of the more complicated approaches available once you've done this. Again, there's lots of resources available if you're interested in learning more, particularly for sort of the principal component analysis and shape tensor topics. We go over them fairly quickly and just talk about really how they're useful. And if you're interested in understanding more about how they work and the model behind them, then you should look into these papers and these documents which show it a little bit better and also show some visualization options you have for data sets like this. Then again, these aspects for models and ROC curves, which we'll review quickly again at the beginning. So if we look at sort of what we covered before, we had the image enhancement in one of the first lectures, and so that was really taking the images from wherever you collect them and trying to process them to clean up the noise and make it represent better the objects that you're trying to extract. The next is the segmentation, which is what we've done in the last two lectures, where you take the image data that you have and you extract it into a number of different phases. So typically it's sort of binary, so it's either a member of this phase or it's not. But occasionally you have sort of multiple phases that you get out of your system. If you're looking at like a clay with different minerals inside it, if you're looking at a car scene and there's pedestrians and trees and streets, then that would be another example of where you would get multiple results out for the segmentation. We talked a little bit about the automatic methods, so how you can process this data automatically and use tools like k-means analysis to visualize and look at it, and hopefully some of you could do that in the exercises. And then a bit about contouring and sort of this machine learning approach. So to kind of review quickly, ROC, because this is really important for everything you do, not just image analysis. So we have this idea of true positives, true negatives, false positive, false negatives. And then we have this curve where we look at sensitivity and or recall and precision, sensitivity, specificity, true positive rate, false positive rate. But basically they're just kind of metrics that condense this true positive and true negative numbers into something meaningful and allow us to sort of evaluate how our system is doing. And I'll show you an example at the end where, I don't know if any of you used the IPython notebook last exercises. You did? Did it go well?
Yes, I think.
Okay.
I'm not so sure. So I did the optimization.
Yeah.
But I'm not sure if it's right already.
Okay, cool. Yeah. So that's the exact sort of thing we try to do. And so with this, you see, you have an image and you try to create an ROC curve from it. And then once you've made this curve, you have a very simple criteria that you're trying to optimize, that you can say very clearly, it doesn't matter what you, or almost it doesn't matter what you do with the image, just try to get this curve up to the upper left corner and get the area under it as big as possible. And then you let the computer adjust all of the different parameters using a variety of different approaches so that you can get this best possible result. And so it means that instead of you trying to focus on it, what you need to focus on is what are these criteria? How can you evaluate it well? And then you let the system search the whole parameter space and try to optimize this curve. And that's really the whole basis for machine learning, deep learning, everything else, is that it's, perhaps a lot of complicated algorithms and approaches, but ultimately what you're trying to do is find the minimum or the maximum of one function. For deep learning, this function then has 100 million variables, but it's just one function and it has one output, and all you're trying to do is either minimize or maximize it. And it's different approaches for trying to do that. And the critical step is really knowing what this function is and what it means, so that when you do find the maximum, you're satisfied with your result. Again, for evaluating models, if you're interested. Here's another example that I thought would be useful to show. This is from breast tissue using differential phase contrast imaging with X-rays to try to find these calcifications better. And so you have sort of this standard image, which is an absorption image, which is what you call a mammography when you go to the hospital and they sort of smash your breast in the system and then shoot X-rays through it. And that's what it looks like. And it's sort of a mess. And it's very difficult to interpret. And there have been many articles about how this is potentially even a waste because it doesn't help you find diseases and it has a lot of false positives. And so it doesn't really give you a good result. The idea with this is to use sort of physics and understandings of how to build systems to get a new kind of information out of it. So rather than just measuring this absorption channel, you can measure this scattering channel as well. And so what you end up with here is that you try to make a segmentation base, or this is your training data, where you know that these are calcifications because someone went through and identified them all as calcifications, and in this case, looked at histology, so sliced open the actual breast and then looked to see if this is actually, this dot there is a calcification or not. And so this then serves as the ground truth for evaluating the true positives and false negatives. You again can take your sort of absorption image and try applying a threshold, and you see that this gives you a pretty terrible result. So a huge number of false negatives and a huge number of false positives, and not very much overlap between the true positives and the false positives, true positives and true negatives. You then can examine the curve. So as you change this threshold, so this was just one value, as you sweep this over the whole range of possible thresholds, or the whole range of thresholds that make sense, you then change these curves. And so as you increase the threshold value, you increase the number of true negatives and decrease the number of false positives, but you also decrease the number of true positives. And so this value needs to be chosen well so that you somehow have an optimum. We now look at this curve and we see that it's pretty terrible. So we would want it to be up here, and in reality it's sort of significantly below that. We then can try the same thing for the scattering channel. And we see that the scattering channel sort of gives us more information. Gives a much better result. And so if we just use absorption or scattering by themselves, it's clear that scattering is a much better piece of information to use because this curve is always, the blue curve is always above the red curve. And so that's a pretty, clear indication that we should be using scattering and not absorption. But then what you can do is you can combine both of the channels together. And so what you can do is rather than just having a threshold for one channel, you can create a whole series of thresholds, so threshold pairs, where you have one threshold value for the absorption and one threshold value for the scattering. And then you can get a much better result than either of them by themselves. And so these are the sort of things that you have to think about and come up with of how it makes sense to combine these data sets together, how you apply a threshold to it, and how you kind of make this curve at the end. And once you've decided that, then you can let everything else be sort of automatically determined. And of course, this is still not great, but it's much better than it was before. Yeah?
How did you integrate the two channels in this case? So you have the threshold for both, and then you combine them into one.
Right. So there's a number of different approaches you can do. So the approach we did in this case was taking the threshold, taking a pair of thresholds rather than a single threshold. And so what you say is if absorption is greater than 0.2 and scattering is less than 0.5, and then you keep all of those values. Another option is to sort of combine the channels together. So you can add or multiply them and then say if the combination of threshold and absorption is lower or greater. And so it really depends a lot on what kind of data you have. And so it's not always, I mean, you have to then go back to your understanding of sort of the physics of the system or the biology and what makes sense to do with these two pieces of information. And so with these, adding them together wouldn't really make sense because absorption is sort of this, absorption coefficient we have, and the phase is then a completely different unit, a completely different scale. And so how you would meaningfully add them together isn't really clear. But having a pair of thresholds makes sense because then you say, if it has a high absorption but a low phase, it's this type of material. And if it has a high absorption and a high phase, then it's this kind of material. Yeah.
What was now the ground truth for your images in this case? Was it like that you actually label images? Or
Yeah. So the ground truth is usually manually labeled things. I mean, so it is very time consuming and intensive to create all of this. I mean, if you look at how people work without techniques like this, they simply have their algorithm and they tweak parameters until they think it looks good, and then they go. And this isn't a very robust method. And this method won't let the computer tweak parameters for you because you don't have any clear goal. You just say, this is when I feel happy about my image, and then you move on. And so with these approaches, you always need to have this defined somewhere. So this ground truth or this labeling. But once you've defined it, it gives you then a lot more freedom with what techniques you can employ. And it makes you more confident that your results are actually meaningful. Because it's not based on your gut feeling, it's based on the analysis you're doing matching the result. Of course, something we'll cover a bit later is sort of this idea, when you're doing sort of more complicated machine learning or more complicated analysis, instead of having one or two parameters, you'll have thousands or millions. And then there's sort of rules for how you go about doing these analyses. Because if you have a million parameters, then you can just teach your system to find these dots exactly and ignore the image completely. But that doesn't help you diagnose breast cancer or identify calcifications. That just helps you match your ground truth. And so this whole area is sort of called overfitting. And there's other techniques you employ once you start dealing with these more complicated systems that sort of prevent you from, making a system that just spits out the answers that you trained it to spit out. Because you want a system that actually identifies these objects. But for the sort of optimization we've done up until now, where you have one parameter and 500 dots to find, the risk of overfitting is rather minimal. But yeah, you can see that you then get a much better result by combining these two pieces of information together. And frequently things like this will come up. I mean, this is what we had in the optimization example here, where you then had to decide you had red, green, and blue channels in your image because this has a depth of 3. And you sort of have to decide what's the best way to meaningfully bring them together. And so in this example, you just add red, green, and blue and divide by 3. And that gives you sort of the average intensity or the perceived intensity as sort of a grayscale image. But if you're trying to identify, white strips on the road, then it might not make sense to use that because you want to sort of focus on objects that are white. And you can use these color channels and the differences between these color channels to evaluate how white something actually is. Similarly, if you're looking for like stripes on the road or if you're trying to find tuk-tuks, then you would be looking for green or yellow. And you can, of course, adjust how you create this sort of combined image so that it matches that better. Cool. So now, kind of when we look at moving on, and the focus of this lecture, it's, what are our goals for doing this? And sort of what the motivations are is that once we've segmented these images, once we've come up with, this is the road and this is the background, how do we now analyze this piece of road image? And so one of the things we want to know is sort of how big is it? Where is it located? Can we say something about shape? How do we sort of compare it? Is the amount of road changing or decreasing? I guess that's not as meaningful with road, but if you're looking at cells, how big are the cells? Are they moving around? Are they changing in size? Are they sort of elongated or stretched? How do we compare two images? And so if you have sort of an image before treatment and an image after treatment for these cells, and you want to show that something changed in their shape or their positions or something, how can you do that? And how can you sort of start to put these things into other approaches? And so once you've taken these images, if you're coming from mechanical engineering or material science or biomechanics, then one of the sort of next steps you have is to bring them into a simulation environment. And so how do you take this image data and bring it into something like a finite element simulation so that you can actually see how the structure reacts in a quasi-realistic environment? Because the information itself isn't that useful. It's the outputs of the simulation that you're interested in. So how do you sort of couple these things with finite elements and other sorts of approaches? Pretty renderings is also an aspect, but one of the things that I'll put in the exercises this time, and it's probably of more interest, is then 3D printing. And so once you've taken a 3D image or you have an object, how do you transform it into something that you can then print out on a 3D printer? Because you can't put image files into a 3D printer. You need to put structures into 3D printers. And fortunately, the structures you need for 3D printers are exactly the same as the ones you need for finite elements and the same ones you need to make pretty renderings. And so it's really about transforming your image from data points to surfaces. That's the challenge that you have there. So in terms of kind of the outline, we talked about the motivation a little bit. Then there'll be the object characterization and sort of the aspects that fall into that, the shape tensor, this ellipsoid representation, and a little bit about scale-free metrics with anisotropy and oblateness, and then this meshing, which is really turning objects into surfaces, marching cubes, isosurfaces, and surface area, and how that's calculated. So we see kind of this slide we had at the beginning. We have our measurements, we did our reconstructions, we went from 56 gigabytes a sample, we did the filtering, enhancement, segmentation, and now we're at 1.7 gigabytes per sample. And so this is still a huge amount of data to analyze, even though it's much smaller than what we had before and much more clearly marked. And so if we look at what we wanted in the first place, it's a single number. And so when you take these images, you want to be able to describe it with a number like volume fraction, cell count, cell stretch, variability, structure thickness, length of road in the image. And so what you need to have is some number you can extract from these images, because the only things you can compare are single numbers. So if you have 20 images and you're trying to compare what changes between these 20 images or what influence does this factor have, the only thing you can look at there is a single number. So you always need to get this to describe your system. So one of the things that's quite important here that builds off of what we've done before, and we used this block actually in the last exercises, if you did the NIME exercises, is this idea of component labeling. And so what component labeling is it's actually an idea that comes from sort of graph theory, is that you have some sort of graph and you want to see if 2 points on this graph are connected. And so clearly if they're directly connected, that's simple. You simply look at the graph and see if the 2 points are connected. But if your question is sort of, is this connected to this? If it's a maze, for example, how do you figure that out? And how do you identify the sort of different components of this that are connected together? And this idea actually fits very well to images, because once you've segmented the images, you have a black and white representation, and you want to be able to see which of these sort of white components or which of these black components, depending on what you're interested in, are connected together. So going back to this image of the road, we see that there's one sort of continuous piece of road in this image, and it would be nice to have a tool that says that this is in fact one component. Because if we look at it now, it's just a list of black and white points. And we can't see from that list if this is connected to that at all. we need to do some sort of further operation in order to check that. And so what comes into this as well is this idea of using a neighborhood. And so with the morphological operations and the contouring, we introduced this idea of a neighborhood, so which pixels or voxels in 3D are connected together. And with component labeling, we have the exact same idea, or connected component labeling. And that's, what's the topology of your system? Do you look for voxels that share faces, voxels that share edges, or voxels that just share vertices? Or do you consider points that are, 2 or 3 voxels away, connected to each other because maybe you have little gaps in your system and you want to fill those gaps in? So one of the areas where that's really important is a lot of sort of brain imaging on animals. You use dyes that you insert into their blood, and it flows around, and then you try to extract sort of the vascular structure of the brain. And the problem you have with a lot of these dyes is that they don't fill in completely, which means you end up with sort of large gaps between vessels. And if you're trying to figure out how well the blood vessel network is connected or how blood flows through the system, then you need to have a system you need to have an idea of which vessels are actually connected together. And if you have a bunch of gaps everywhere, then that doesn't work well. So if we look at sort of what's the algorithm behind connected component labeling, it's a sort of brush fire style algorithm, or the simplest implementation that we can make is one of these algorithms. And so what you do is you take each point and you sort of look at all of the neighbors of this point, and if the point is a neighbor of this point, then you set it to be part of the same class. And so you perform this iteratively until you've sort of covered all of the points in the image. And so this is after one iteration. You have your begin and that your pixel grown is here because this is part of the same group. And then you perform the same step again. And so this is what it looks like after 10 iterations. This is what it looks like 10 iterations from if you start growing at the end instead. So here we started growing at the bottom. There we started growing at the top. And here you can sort of see how this progresses. And you can do the same thing for an image. And so you have an image here. It doesn't render correctly. But you have an image with a number of different colors there. I think the way the image looks, maybe I can fix it now. Okay, so now it looks correct. So that basically you have an image, and so you have all of these points labeled with different colors. And so the points down here are sort of 0, and the points sort of increase 1, 2, 3, 4, 5, 6, 7, 8. And what you're trying to do is look at this image and see which of these regions are connected together. So of course, when you look at it with your eye, you see very quickly that there's sort of a gap. There's a triangle here or there's a cross here where the components aren't connected anymore. And so that reasonably, you'd expect 4 different components to come out. We then explicitly show the topology here so that you see each point is connected with the point above and to the right of it. So these diagonal points are not connected, and you can see all the different lines. And so what you do here is you check for each point the neighborhood of it, or first you give each point its own label. And so that's where you say basically the label is the position, and that forces each point to have a unique ID. You then start with each point, and you can see for all its neighbors to take the minimum label in that group and iterate this until no labels are changed anymore. And so you can see after 2 iterations that you've already grown out quite a bit, 3 iterations, and by 4 iterations, actually all of the points are now in these labels, and you only have 4 distinct groups left. And so it really lets you see how this progresses and how the number of labels decreases and how an algorithm like this works. Of course, this is a very naive implementation, and on your computer, or in programs, these are implemented much more efficiently. But I think it's helpful to kind of see how these algorithms work, because this is one of the very important ones. It's usually sort of the first step you take once you do the post-processing of your images, because identifying the number of different components and how they're connected together and how they look is one of the main things you extract from these images. So we can now look at sort of how this looks for a real image, and that you can then take out the image we were looking at before, or in one of the earlier lectures, where you had these cells. You can then apply this connected component labeling, and now you have all of these cells labeled differently. And this is then very helpful, because now for each of these cells, you can calculate their area or their size by looking at the number of pixels inside. And so by counting the number of pixels in each one of these groups, you can then see what the size distribution looks like. And this is actually a very helpful criteria for defining what's a cell and what's not a cell. And so now we can see, there's a pretty large distribution between 0 and 75, maybe square micrometers. And if we exclude the points below 25, that gets rid of a lot of the dust. And so that rather than trying to filter this image better, we can simply take the component labeling and say all these really tiny objects we want to completely ignore. And so by doing this, it gives us the ability to get rid of those objects, so we can just show the larger objects and keep those. Yeah. And so now we can sort of perform these next steps in analysis. And so we can count the number of voxels inside each image to estimate the volume. We can look at the change in volume. We can analyze a number of other parameters. And this week and next, we'll cover sort of the specific things we can do with this. So if we go from the labels, we now have a labeled image as well as the cells. The first thing we want to do is sort of count the cells. This is quite easy. This is just the number of unique labels you have, or the number of cells you have in the image. And so it doesn't require any additional looking at this image. You just look at the number of unique labels you have. You now want to be able to say something about the cells and start to compare them. And so what do you do for this? So the first thing we can do is we can measure the center of volume. So you can ignore, for whatever reason, these lines show up after every equation, and I'm not really sure why. I think something with LaTeX or MathJax is screwing up, but you can ignore these lines completely. They don't have any significance. It's not some sort of equation or something like that. So now we basically have each object that we're looking at. So this is then one of the cells that we had in this image here that we focus on. And we have kind of a list of points. And what we want to do is we want to define where's the center for this point. And we choose, the center of volume for this. And so this is the weighted average of all the points that we have in the image that are inside this given point. And so that gives us this value. This is, of course, important to define because for a lot of things you're interested in center of mass and not center of volume. And so here we just looked at all the points in the image and we found the average of all of those points. The center of mass then looks at some other piece of information, potentially the intensity that came in, or some other channel you have that gives you some density information, where you can then calculate the center of mass and compare this to the center of volume. And so we see in sort of a cell like this, if there's a gradient across it, then the center of mass is actually shifted. So the center of mass would be sort of up to the right, while the center of volume is in the center. Calculating the center of mass is quite easy. You just use this weighting function, and you multiply it by all the points in your image, and it's just a weighted sum for x, y, and z, or however many axes you have. To now do extents of an object, this is fairly easy, particularly once you have the center of mass. You can basically just take all the points that fall in your image, and you find the maximum value minus the minimum value in each direction. And so that gives you a lot of information about the size, but it's not really a physically meaningful metric because no one asks you how big your cell is in the x direction or the y direction. They ask you how big your cell is in general, or how long is the longest axis of your cell. And so does anyone have any ideas how you would go about calculating the longest axis? Yeah? That would be the most complicated approach. I mean, that's the one we ultimately use. Yeah. Usually people guess something slightly simpler, like rotating the object around and measuring the extents until the extents are highest, and then you know the length and the longest direction. Of course, that's not very efficient, but it is sort of intuitive of what you're doing with the sample. PCA, I think, is... has everyone heard of principal component analysis before? And has everyone used it? Okay. I mean, we'll go over it briefly, but I mean, it's a quite useful technique. Cool. So now, what we'll try to define is that once you have sort of these measures like extents, how would you go about calculating some meaningful comparisons out of them? And so one of the most frequent analyses that you do in terms of shape is looking at anisotropy. And so it really is about saying how sort of oriented or how asymmetrical your object is. That if it's the same in every direction, it would be isotropic. And if it's not, then you want to be able to sort of describe this some way. And so is it tall and skinny, short and fat, or perfectly round? And so because the definition of anisotropy is very vague, there are a number of different ways to calculate it. And if you look through scientific literature, you find all of these and many, many more. And it seems that kind of each person defines this whatever way makes their results look the most exciting, and not really based on a solid understanding of what parameters they're extracting or what they might be useful for. And so, of course, the basic idea is that you compare the longest side to the shortest side. Or here you have the average side length. But what exactly you do to it isn't really well defined. And so what we'll try to cover is what differences choosing different metrics have, and what metrics I, for myself, decided were the best. But of course, your criteria are probably different, and everyone seems to come up with a different result for this. And so I think what the best thing to do here is that you make a bunch of sample objects, ideally objects that somehow represent what you're trying to look at. And so if we're looking at cells that are getting stretched out, then this would be a pretty good model for those cells. That this would be something that's not stretched out at all, and then it progressively gets more and more stretched out till you can sort of barely see it. It's so stretched out. And so here we have sort of the size of the sample in y, or the y extent, and then these 4 different anisotropy values calculated for each one of those stretches. And so It's a little bit confusing because 0 there. So this is the bottom one there. This is the next to the bottom one, and so forth. And so for the top sample, all of them are in complete agreement. So if you put a circle into any of these equations, they all give you 0, which is what you'd expect. If it didn't give you 0, it wouldn't be a very good indicator of anisotropy. The difference is how these values change as you sort of stretch your object out. And so here, when you go to sort of 4, 3, 2, 1, you see that this first number starts to get very big very quickly. And so by the last value here, you have an anisotropy of 5,000. And with these two other ones in the middle, you have an anisotropy of 1, 1, and 2. And so these ones sort of have a more bounded nature. But if this is something you're really interested in finding, then using the first measure of anisotropy will then give you a very large value when that's very small. And so if you're looking at something where the orientation is very important, or where you want to be very sensitive to these really strongly oriented objects, then using something like this would make sense. Of course, the problem with a method like this is when you start to do averages, that if you have one sample that's like this, and you have a value of 5,000 for that anisotropy or even higher, then you can end up having that sort of skew your whole distribution and make it very difficult to figure out what kind of the average shape of your objects are. And so here we sort of see that as curves. We go from 1 to 8 again, and we see how these different curves behave. And so this is the first one where it goes from basically infinite down to 0, and these other curves which sort of fall in between them. We then take sort of randomly shaped objects. And so here we have a list of objects that are more or less randomly shaped, where they have an even density distribution in x and y. And so we just make something where there's a fairly random distribution of everything. And now we can show sort of what are the values of the anisotropy compared to each other. And so we see that these curves now look different again. And this is the scatter plot matrix, which shows you sort of the first one versus the second one, the first one versus the third one, the first one versus the fourth one. And then this value down here where you see the distribution of anisotropy for all of the samples that you're looking at. And so here you see that these curves do look quite different. And which of these curves best represents what you're trying to see in your image. And so again, it's about trying, if you have an even distribution in the lengths of your objects, then do you want to have an even or mostly even distribution in your anisotropy or not? So that's kind of the factors that you use to pick which one of these terms makes the most sense. Because ultimately, once you've decided on this metric for anisotropy, it will affect the rest of your results. And so if it's not matched well to the system you're looking at or the experiments that you're doing, then you can get sort of very strange or not as reliable results as you would ideally want to get in your system. Cool. So now we can sort of do extents with all of the objects so that we take all of the cells we have in this image and we calculate for each of them sort of what the extents are in each direction, and we show them as little lines on this plot. And then we can show each one of the cells in a different box where we see where the center of volume is and what the extents look like for that image. And I think this is quite a helpful plot to make, because when you calculate all these metrics, it's important to see how these metrics are actually reflecting your current samples. And so when you have, a sample like this sort of violin-shaped object, you see that the extents don't match well at all because this object sort of rotated at 45 degrees. You also see that there's some sort of object like this that kind of has this loop inside of it, and that's also probably not being represented very well by this simple extent model. And so these objects are mostly round, but there's quite a few which aren't round. And there's actually a few like this one where it's probably 3 different objects connected together, and that's why it has such an interesting sort of structure to it. So I think we'll take a short break now and then continue with the rest afterwards.